before, what is Bay Area SfN Youth? We're basically a community of high school students from schools all across the Bay Area, connected by an interest in neuroscience. We're a student-run organization, and our goal is basically to promote awareness about neuroscience in the brain and many basically neurological conditions to our community, and to share various aspects of the broad field of neuroscience with the help of guest speakers, interactive activities, neuroscience competitions, seminars, dissections, walks, and so many more engaged events. And so we'll start off by summarizing the video from last year. Hope you guys can see it. A lot of our community engagement events, so brain dissection, volunteer events like walk, community speakers as well. And that's what SfN really does. Our goal is to promote awareness of the brain and neuroscience throughout our entire community, especially in the Bay Area. And so who are we? So this is our core team. I'll start by introducing myself. I'm Shabak. I'm a senior, and I'm a co-president along with Harish. And our goal is to make sure that SfN is running smoothly, to set up all these events, and to make sure we're able to get outreach and promote the community. Harish, you want to go next?
Yeah, so I'm Harish. I'm also a senior at North High School. So this year, yeah, we have a lot of committees, and in the committees, I'm leading the newsletter committee. So,
Levi, you want to go next?
Hi everyone, I'm Eva, and I'm the Senior Vice President of SfN, and so yeah, I'm head of social media and promotions for as far as our committees go, and I just kind of try and keep everyone on track for deadlines and stuff like that.
Hi everyone, my name is Ritika. I am a senior at Saratoga High School, and I'm the Vice President of Special Events. I am also the lead on the website development committee, so that means a lot of just working on the website and redoing things, and my role as Vice President of Special Events is mainly working on our really big event, which is in the spring, called SfN Wonder, and organizing things for that.
Alexander.
All right, hello everyone. I'm Alex, a junior at Lindbergh High School, and I'll be serving as SfN secretary this year. And I'm also the co-lead with Harish for the newsletter committee. And as the secretary, I'm responsible for creating meeting minutes and overall just organizing the SfN Google Drive.
Hi everyone, I'm Suman. I'm a senior at the Harker School, and I'm SfN's treasurer, and I'm also the lead of the fundraising committee, and I make sure we have all the funds for all of our events.
Simran?
Hi everyone, I'm Simran. I'm the public relations officer this year, and I'm the co-head of the social media and promotions committee. So my job is to basically create all the promotional materials and handle outreach and our social media.
Diana?
Hi guys, my name is Diana. I'm a senior at Lee. I am the co-student representative liaison and the co-lead for the neuroscience class committee. And I'm interested in computational neuroscience and biology.
And Netra?
Hi guys, I'm Nathra. I'm also a student representative liaison and co-lead for the neuroscience committee along with Diana. So our role is to basically be the bridge between the core team and the student representatives. So if any of the student representatives have any questions about their roles, then they come to us. We remind them on their deadlines, etc. And in the neuroscience class, we're basically the leaders for the class itself and all of the teachers that are part of our committee.
That's us. That's a very SfN core team. But all the work that we do would not be able to be done without our amazing student reps who really help us get the communities running, get the organization running, and are really responsible for all the promotion and outreach that we can actually have our events happen. So once again, thank you to all our reps who are attending and who've been helping us throughout the year to help set up these events. So Harish, do you want to recap the 2019-2020 SfN?
Yeah, so 2019-2020 SfN was a great year. We first started off with the August brain section, which is kind of an annual event that we do, which gets a lot of attention because it's really nice. And also in the September 2019 meeting, we talked about it was a speaker event, and we talked about trauma. An amazing Dr. Raina Sangui, she gave a presentation about trauma. And in October, we did our walk to end Alzheimer's. Alzheimer's is something that we're really committed about, so we participated in that event. So in December, it was a dual speaker event. We talked about mindfulness and just stress in general, the finals. So January, we did the Neuro Bowl in preparation for the Brain Bee, which is an event that happens in late January. And in February, we had an internship workshop, which well done summer internships for high school students.
Yeah, and so these are basically a summary of all our events this year. Normally, we do have an SfN Wonder, which is like a big cumulative event in March, which kind of has a lot of speakers from various different fields in neuroscience, because neuroscience is a really broad field. Come and share their research and some of the stuff that they've done in neuroscience as well. Unfortunately, due to COVID-19, we were unable to hold that event last year, but we look forward to being able to hold that event this year in March again. And so just a couple of changes to SfN this year for many of you who were there in previous years. We're going to have more student reps involved, because and this year we already have the committee system, student reps are running. We're also going to involve in more promotion so we can have greater events. And especially because this year is our first year going online due to COVID-19, we're not able to host in-person events. We're gonna have a new focus on our community, trying to help up our community as well, in addition to just having speaker and presentations event. And we'll vary up our schedule with more interactive activities. And so this is our first online event, and we're really glad to have all of you guys here, and we look forward to getting it started. And so just a bit before we get into the actual presentation, there's a bit of a sharing of our schedule of the year. So right now in August, we're having our speaker symposium. But in September, we're thinking of having a medical movie mystery along with those speakers when they talk about the movie event. In October, we're going to have the Walk to Alzheimer's. Although it's going to be a bit more like restricted this year due to COVID-19, we're currently discussing them to find ways to have smaller groups of individuals walk as we fundraise against Alzheimer's disease. In November, we're going to have another guest speaker event, and in December, it'll be a guest speaker event plus an interactive activity. In January, we're going to host our dual event month where we host a Neuro Bowl to help people prepare for the Brain Bee, and also have an internship workshop as well, where we kind of share basically some of our research and talk to high schoolers about opportunities of gaining internships in high school. In February, we have another guest speaker event, and assuming that everything starts to settle down, hopefully by March, we'll be able to have our SfN Wonder, which will kind of culminate the Brain Awareness Week, which is in middle March. And then in April, we'll continue our special needs volunteering as how we helped out with senior shelter volunteering last year. We're planning on continuing with that in our April event. And so now we want to introduce Dr. Joseph Parvizi. I do think he's having a bit of technical difficulties and hasn't been able to join the room yet. So we'll have a 1 to 2 minute delay, try to figure out what's happening with Dr. Parvizi, and we'll come back in 2 minutes to start our event. So stay tuned just for a couple minutes. We also may start with a Kahoot as well if these technical difficulties do continue. So just give us a minute or two, and then we'll get started with our event.
And just a reminder for those that came late that we have our check-in form posted in the group chat. So if you guys can please fill that form out so we can take your attendance, especially important for the student representatives to fill it out, that would be great.
We'll get started with Kahoot in a minute. We'll start the game now. Different kind of topics that our speakers will talk about. We'll start off with a little yeah, there's quite a few instances of trauma due for female veterans, especially which can cause non-combat related TBI. And I think one of our speakers will definitely cover this topic. Great, yeah, there is, PTSD has been shown to correlate to higher rates of hypomethylation, less methylation in the glucocorticoid receptor gap, which we'll also cover in a bit. Theta, right? There are 4 types of brain waves, and theta are the ones that are most often sleep, but are really dominant during deep meditation as well.
Wait, is anyone recording this?
Yeah, we're recording it. Right, gamma, they are the highest frequency waves, and they're above 35 hertz. And there's a lot of people are interested in neuroscience and know quite a bit about the brain. It's great. Okay, so there's 2 answers actually. The common non-cause such as TBI, confused seizures, and apoptosis and excitotoxicity, both really severe effects of traumatic brain injury, TBI. neurotransmitter GABA is one for sleep process, so it's kind of like the inhibitory neurotransmitter. Great. Right, it can lower ICP and improve cerebral perfusion. So female veterans are ones who are most likely to experience post-concussion syndrome. We have a couple more questions, and one of our speakers is showing up, so we'll get started with that as soon as he finishes.
I'm sorry for being late. I'm the counselor for programming here.
No worries.
Great, we have the last couple questions left. Yeah, most of you have got it. Epilepsy. Perfect. Right, so three of them, right? There's petite mal, which is like localized, grand mal, bigger seizures, and focal epilepsy as well, which will also be discussed really soon. Three more questions. Right, wow, there's a lot of cases, 125 to 180,000 new cases of epilepsy every year. So we're into our last couple of questions now, 13, 14, and 15. Excellent. Yes, grand mal must be generalized tonic. They're both be similar names, but generalized is grand mal epilepsy. muscles in the arms, legs, and back stiffening, and it's around 20 cycles left to seizures. They tend not to be too long lasting in terms of actually having effects.
If I may add, if I'm not mistaken, after a seizure, it can often result in tiredness or confusion.
Right. And the last question. Great, yeah. The feet loss does affect the cortical system. And so for our top three Kahoots, it was pretty tough. So congratulations, Natasha. So good job, everyone. And that was our Kahoot. And now give me 1 second. Great job to everyone who participated. That was a pretty tough Kahoot. And now we're just gonna have 1 minute as our next speaker is joining right now, Dr. Adamson. And so let's share the slides so we can introduce our next guest speaker, or our first guest speaker, Dr. Adamson. Is everyone able to see my screen? Sure. There we go. So our next speaker will be Dr. Adamson. Dr. Adamson, are you here?
Yes, I am. Can you hear me?
I can hear you. Wonderful. And so, Netra, would you want to introduce our speaker?
Sure. So Dr. Adamson is a clinical associate professor of neurosurgery at Stanford School of Medicine. She's a senior scientific director for the Defense and Veterans Brain Injury Center, and she's a principal investigator and site PI on numerous neuromodulation clinical trials under the Department of Veteran Affairs and Department of Defense-funded grants, which is very impressive. So for her education, she obtained a BS in neurobiology and women's studies at UC Irvine, a PhD in neuroscience from USC, and a fellowship in psychiatry and behavioral sciences at the Stanford School of Medicine. So some of her accomplishments is that she authored numerous peer-reviewed publications on the cognitive and neural basis of Alzheimer's disease and on a wide range of topics in traumatic brain injury. She's a leader in identifying gender differences in brain injury, particularly in the veteran population, and she's received national and international recognition for her work. So today she will discuss traumatic brain injury, otherwise known as TBI, the neural basis of the Alzheimer's, and the gender differences in brain injury. So without further ado, we present Dr. Adamson.
So should I share my screen, or are you going to do it from the presentation I sent earlier?
I think you could share your screen. That might be easier for you as well, like when you're transitioning.
Okay, sounds good. I will do that in just 1 second. How
guys,
is everyone doing?
We're doing good. How are you?
Doing okay. Doing okay. So let's see. Got two screens. So hopefully we will make this.
And guys, if you have any questions, we can type them in chat. I think Dr. Adamson, that should be fine, right? If you're able to type in chat, or we can also ask them at the end of the presentation, whatever's convenient for you.
I think end of the presentation would be best.
Sounds good.
Okay, how does that look, guys? Does that look good?
Yep, we're able to see your screen.
Yep, looks good.
Okay, that's perfect. Okay, so thank you for the nice introduction. Did you guys have a chance to hear Dr. Parvezi?
We're having some technical difficulties, so Dr. Parvezi wasn't able to join.
Okay, and you have another talk after this? Okay, wonderful. So thank you very much for joining, and I'm looking forward to having great questions from you guys. Thank you for the introduction. I'm going to tell you guys a little bit about my job right now. So I'm the research director for the rehab department, which is basically looking at a lot of patients who have traumatic brain injury, amputations, spinal cord injury, a lot of things that have to do with the brain and spinal cord, and then figuring out the entire spectrum of, using epidemiology to figure out what the incidence is, and then figuring out how to the diagnosis, and then on top of that, figuring out how to treat them. And the rehab really comes in and how to end up treating them, and then make sure that they reach uh, some level of the performance that they had before they got injured. So you can see that there's a lot of interaction of all kinds of disciplines in this. I'm also clinical associate professor in neurosurgery at Stanford School of Medicine. I'm a basic neuroscientist from training, and have been at it for about 15 to 16 years. So what I'm going to talk to you about today is a small but very important part of my work, and I thought it would be very interesting because it kind of ties in with a lot of stuff that's been happening with the COVID-19 pandemic. And with the COVID-19 pandemic, we have really realized that there are a lot of health disparities and healthcare disparities. And one of the biggest ones is the fact that we need to apply precision medicine to certain groups of people that are disadvantaged. And one of the groups that are disadvantaged is in the female minority. So today we're going to talk about biology and the environment, and I tried to make this interesting. So we'll go to understanding the female brain. So first, I wanted to say that as a presenter, I have no interest to disclose. I do represent both VA Palo Alto and Stanford, but these are just my opinions, and it's you can blame me for whatever I say. But today we're going to talk a little bit about the basic anatomy of the brain. And if I was in front of you, I would say, tell me where the amygdala is. And you'll raise your hand and you'll say, well, there it is. But we can't do that because we're just to disclose. I do represent both VA Palo Alto and Stanford, but these are just my opinions. And it's, you can blame me for whatever I say. So today we're going to talk a little bit about the basic anatomy of the brain. And if I was in front of you, I would say, tell me where the amygdala is. And you'll raise your hand and you'll say, well, there it is. But we can't do that because we're virtual. So but we're going to talk a little bit about a different version of this, is how science has changed from 1950s. And this is an important part. We're going to talk about the sex differences in behavior as well, because the brain, unlike any other organ, actually changes the way you perceive yourself and the way you perceive others. And it also affects your behavior. Sex differences in the brain as well. We'll talk about the health impact of understanding what the sex differences are, both in brain and behavior. And we'll talk about leadership impacts, how it would impact you dealing with your boss and having the leadership be represented. And then we'll talk about a little bit more of take-home messages. So the first thing is wanted to give this quote from Darwin. As you all know, we all studied this in high school and even earlier about Darwin is very famous. And this is his quote: "The chief distinction in the intellectual powers of the two sexes is shown by man attaining to a higher eminence in whatever he takes up than woman can attain, whether requiring deep thought, reason, or imagination, or merely the use of the senses and hands." So you can understand that while society has moved past many outdated Victorian mindsets, the legacy of sexism in science remains. And the reason why I also say that is most male scientists conveniently choose to ignore that women were actively denied access to higher education. So the University of Oxford didn't admit women as full members until 1920. They were excluded from great scientific academies. So you would call it, Marie Curie was denied membership into France's Academy of Sciences in 1911, even though she was a Nobel laureate. So these are just some things that kind of tie the inherent biases that go from science into leadership, into how the world perceives women as scientists.
Then you move forward into what popular depictions are. Popular depictions for male and female, I'm sure you've seen this, and it's constantly. I just got another video yesterday on some WhatsApp group, and it talks about how there's male and female brain differences, and this is what a male brain looks like, and this is what a female brain looks like. Well, the scientific depictions are slightly different. So this is, as some of you may know, has to do with brain connectivity. So we are incredibly advanced in looking at brain imaging. So we can not just look at the structure of the brain, we can also look at the function of the brain. The brain is not just gray and white matter and those wonderful CT scans that you get or the MRI scans. You can actually figure out how blood flows in the brain, and you can figure out how the different connectivity patterns in different connect different parts of the brain, and how those connectivity patterns are different, not just by disease, but inherently in everybody between men and women. So if you read this a little bit, it says a new way of showing the connectivity of the brain is called the connectome, which reveals significant differences between men and women. Now, the difference doesn't mean that one is lower than the other or one is higher than the other. It's just different. So a typical male brain, this is based on a lot of different, lots and lots of data. So it's templates and created with hundreds of patients or scans. The typical male brain, most connections run between the front and back parts of the same brain hemisphere. So one hemisphere, which could account for the better spatial skills and motor muscle control in men. So we'll talk about that. That goes into the behavior part. When I say behavior, I don't particularly mean psychological behavior. I also mean like any of the outputs that you have that are motor or other skills that can be done. A typical female brain, however, many more neural connections go from side to side across, so what we call bilateral as opposed to ipsilateral. So left and right hemispheres communicate. Scientists think this could account for women's better verbal skills and intuitive abilities. I don't know what that means, but anyways, I need a little bit more objective and understanding of what intuitive would mean here. So you move forward to this kind of, depiction, the connectome. It's very nice, but it does lead to some kind of problematic research. So sex differences are based in animal studies. I'm sure you guys know that already. So those animal studies are 99% male species. There's a problem with that. There's a problem with basing your data on animal studies. The biggest problem is a rat brain or a mouse brain, approximately 71 million neurons. This is the human brain. It's 86 billion neurons, and it's 16 billion just in the cerebral cortex. So the human brain actually weighs 1.5 kilograms, but only 2% of the whole body weight, which means it uses 25% of your energy. It's a very greedy organ. So you can see it's very difficult for the human brain with all its wonderful, gyri and sulci to be compared to something that is animal, because you can see it's completely smooth. So when you have a piece of paper, when you have a line or a piece of paper, you draw a line on a piece of paper. I'm sure you've heard this example, and then one end doesn't meet the other. But if you fold it, those two points can meet, right? And that's what's happening in the brain. So you're creating a lot of what's called associative cortices, and that really makes a huge difference. So you're like, okay, well, what's the big deal about this? Well, there's two schools of thought, and Stanford's pretty big in this. They've done a lot of research in sex and gender. So sex versus gender are two different words. Sex, when I use sex, it means what you're born with in terms of male, female, transgender, and all the different types of biological way of defining what your sex is. Gender is a little bit more environmental, and it's also what you define yourself as. So if you look at gender, it'll be any between sex differences and thinking abilities are due, and those lead to two thoughts, right? One is that any between sex differences and thinking abilities are due to socialization practices, artifacts and mistakes in the research, and bias and prejudice. So everything is environmental driven. Biology is biological basis of sex-based cognitive differences. So there's a lot of data from animal, cultural, and neuroimaging studies that say that, well, biology really drives it. Now, one of I'm sure maybe some of you have heard of this, is that they've done some animal studies in rhesus monkeys in which males strongly preferred toys with wheels over plush toys, whereas females found plush toys much more likable. Would be tough to argue that the monkey's parents bought them, sex type toys, or that some society encourages with male offsprings to play more with trucks. And then a much more recent study established that boys and girls 9 to 17 months old, an age when children show few, if any, signs of recognizing either their own or other children's sex, nonetheless show marked differences in their preference for stereotypical male versus stereotypical female toys. So the biology part of it is like, okay, so there is definitely a difference at that point. So there's two schools of thought. I don't believe in that those two schools of thought exist in their own. There's definitely an interaction, and there's definitely something in the middle. But we'll get to that in a little bit. The next slide kind of takes you from the brain part into the behavioral. And this is actually very interesting, and it helps us understand it as we move into psychology, as we move into leadership, as we move into health impact, biology, healthcare. So from the biology, we move into what's called verbal abilities exceed that of men in women. You've heard that a lot. You're like, women talk too much, or oh, they're easier to express themselves. Well, there's some truth in it. Some studies have shown that women are reading comprehension and writing seems to be a little bit better than men, and this also depends on age and depends on your IQ and depends on how much education you've had. Your perceptual speed and fine motor coordination is also better. Retrieving information from long-term studies. This is all based on a plethora of studies. But what are men good at? Men are good at superior visual skills that exceed that of men. Now, again, this is talking about aggregate data, average data. So we'll talk about other ways of looking at it. Men are better at tracking objects in 2D, 3D spaces. Navigation skills are distance and direction based as opposed to landmarks. So I know for myself, I'm pretty average at this. I know that I am much better figuring out directions when you tell me, oh, it's right next to this convenience store or it's right next to the school, as opposed to looking at, directions that are displayed with this, you have to go 2.5 miles and turn left and things like that. So those are the differences. This is what people have found in previous studies. So you're like, okay, what does neuroscience tell us if we go by this behavioral stuff? Well, like I said, we can do a lot of imaging now, and we can actually do MRI that's gray and white matter. We can do MRI that's just top layer, so you can actually segment the brain with different parts of the lobe. You can actually talk about blood response, so you can actually map the way your brain is supplying blood to its different areas. So you can look at BOLD, which is blood oxygen level dependent response. You can also do what's called white matter tracks, which is looking at how water flows in the brain and figuring out how the integrity of the fibers is. And this is actually a depiction of all the white matter tracks in your brain. So this actually comes in handy when you're looking at brain injury, how your axons actually break, and from the actual breaking of the axons, how that can lead to any of these types of imaging to be not at their best. So you can see injury through looking at all these different types of imaging. You can also see this under the microscope in much easier way, but I'm just talking about imaging. So brain differences in terms of, figuring out what the differences are, how it turns into behavioral differences, and with all the technology that we have now, we've really figured out that size does not matter. And it's a very interesting thing to say, because it's a popular culture thing to say. Size does not matter. And while the genes that are hosted, see, I'm combining a lot of different types of biology terms in order to address this huge topic about sex differences in the brain, because it's not just going to be talking about the brain, it's going to talk about genetics, behavior, all of these things coming together. So what's really interesting is that while the genes hosted on the X chromosome and the Y chromosome So there's 27 on the Y chromosome and there's 1500 on the X chromosome. They may once have had counterparts on the other. That's not now the case for only a few of them. So every cell in a man's body, including his brain, has a slightly different set of functioning sex chromosome genes from those operating in a woman's. Adjusted for total brain size, which is brain size for men is bigger, it's a woman's hippocampus, for example, which is very critical to learning and memorization, is larger than a man's, and it works differently. So conversely, it's very interesting. A man's amygdala, which is supposed to be the area where you experience emotions, recollect emotional experience, is actually bigger than a woman. So total brain size, again, is bigger in men. Hippocampus is bigger in women, and amygdala is bigger in men. So but in behavior, women respond more to emotionally disturbing content, more vivid and more intense. So that's even though they're generally, on average, amygdala is smaller in women. So again, I'm showing that figure that this kind of connectivity and looking at structural differences do make a difference in understanding how behavior maps onto the brain. The two hemispheres of a woman brain talk to each other more than men's do. So now we move further to figuring out, you're like, okay, well, these are the differences. Now we've understood what the brain is and how the behavior is and how it maps, and there are the differences, and we're kind of working on that. How do those differences actually translate into when you're actually in the hospital and trying to figure out disease characteristics, or you're a researcher who's trying to study disease characteristics? Do these things even matter? Yes, they do. So here's the difference. You've got the male brain and you've got the female brain. And we've actually talked about why it's different, right? So you've got optimized for motor skills, front to back connections, more ipsilateral, 10% larger. And the female brain, it's stronger side to side connection, optimized for intuitive thinking, verbal learning, and more gray matter as opposed to the female brain. This is actually from Northwestern University's research. What are the possible health impacts of that? The male brain is more likely to develop alcohol dependence. It's 3 times more likely to be diagnosed with antisocial personality disorder, 4 times more likely to have autism, twice as likely to develop Parkinson's disease. See how this is all written from the perspective of previous incidents and studies, right? Possible health impact in the female brain: twice as likely to have depression, twice as likely to develop Alzheimer's disease in the US, 4 times more likely to develop multiple sclerosis, more likely to have a stroke. So how did we go from behavior and all these brain differences, and now look at this. This is all public health literature. This is based on numbers. And now you have to figure out how to understand the mechanisms by which this is all actually working. Well, let's think about how a woman's body actually works with medicine and how with medications, and how a man's body works with medication. It's actually very interesting. Prescription painkillers, women get more relief, but they have a harder time with addiction. Men overdose more. Do you know why that is? Again, I would just about talk and wait for you guys to answer questions afterwards. It's because women they don't digest the medicine fast enough. So it stays in their system longer. So they get faster relief, but they have a harder time with addiction. Men, they overdose because it's not affecting them as fast, so they take more. Antidepressants, some of them are more potent for women. Less acidic stomach, more absorption, interaction with other drugs. Zoloft is better in women, tricyclics better in men. Sleep aids, liver breakdown is faster in men than women. If it's faster, you're going to take more of it. And if it stays in your system longer, you're going to get more addicted to it. Anti-anxiety, stomach absorption, liver breakdown, same more potent effect. Antipsychotics, liver enzymes work at different speeds in men and women, which might affect how the body responds to antidepressants and I don't know how to pronounce that, painkillers and other drugs. So that's actually from, I took it from Scientific American and then went and looked for some other papers that were very specific to this. Now, How does all that impact social interaction and leadership? So this is, kind of moving from biology all the way to the environment. So now, as you know, as in the case of autism, science has expanded our minds. Like before, it was just like one category. Now it's a huge spectrum of disease. We know that some people have very mild versions of it, and some people have very strong versions of it. And that seems to be happening to a lot of diseases. So as a scientist, I don't like categorizing things. I like continuous variables so that I can see different varying within a disease, you can see a continuum. So the categorical understanding of what autism was or what depression was has changed into it is a continuum. You can be really on the one side versus you can be on the other side. And it will make a very important, the reason I'm bringing this up is because we're going to tie it in as how we understand the difference between men and women in the brain. So our environment has changed. Leadership has more women in it. We're still very different from men in a way that is not at all categorical. Leadership gap is decreasing very slowly, but women held 6.6% of Fortune's 500 CEO positions in 2019. And as you know, just as of last week, we have a VP that is a VP candidate that is a woman. So leadership is changing, and the way women lead is not going to be the same as the way a man leads. And all these things that we learn about how different we are in good or bad, doesn't matter. How different we are helps us understand how to deal with the environment, how to make an impact, and how to understand each other despite any of the differences that may exist and that society has kind of put in our head that we are different and all that stuff. So how does that affect policy? So this is National Institute of Health and their policy on gender. They really realized that this is a really big deal about 10 years ago. And they started coming out with white papers and started really, now it's really important to address gender and sex in your research. So as a scientist, when I write grants and I get funded and I'm doing research, I have to really make sure that I address the disparity that we see in women and include, make an extra effort in recruitment to bring women in so I can study them. So I'm not doing, just male rats or just males. And it's particularly very difficult when you are actually studying veterans or in the military. So this may be the case across the world, and it's changing a little bit. Women are entering military a little bit more than before. But they came up in 2013, an Institute of Medicine report on returning home from Iraq and Afghanistan, and they found that the women who were at that time in the military, they were facing unique stressors that were different from men. And that's what, VA and NIH and CDC and WHO, they started thinking that, the medicine you give the men, the way you treat the men is not going to be particularly valuable in the way you treat women. They have a very specific response, as you know from the previous slides. And they were like, you need to look at this. We need to really figure this out, because otherwise we're going to have this huge population of women that is not, that's going to be underserved. And that really is what has happened. I just finish doing a COVID-19 survey across the world, and it's under review right now. And across the world, in that 1 month of quarantine, women were under way more stress than men across the world. Germany, Pakistan, India, Mexico, UK, Canada, US. So it's really important and why is that? We can talk endlessly about why that is, but we need to actually do research in it and write about it and make sure that it's addressed as we move along. So why do I do this research? So I got a paper recently published. Was that recently? What's the date on this? I forgot. I think it's end of 2019 or beginning of 2020. Since then, we've had a couple of more papers that have been published and accepted in this topic. So we looked at sex differences in symptom presentation, what they are reporting what they are reporting in terms of their symptoms, and whether they have any kind of effect of those symptoms in their functional outcomes every day. And we looked at this in a matched samples of veterans in which you took 30 men and 30 women who had brain injury and 10 years ago or 5 years ago. And we really looked at whether men were reporting more symptoms, what kind of symptoms they were, whether women were reporting more symptoms or what kind of symptoms they were. And if reporting of those symptoms, I can say I have a headache, it doesn't matter until it affects my daily work, right? So if I'm like, oh, I have a headache, that's great. I mean, I can take, nobody cares. But if it's not helping me do my work and it's actually not letting me go back to work, not letting me get an education, not letting me get a job, then it's what's called a functional outcome. It's an actual thing that's affecting the lifestyle. So we did this and we found that women have more cognitive problems than men after brain injury. These are military women. They are actually showing more psychiatric problems. They also have more vertigo. And there seems to, and it's after years of injury, and there's a huge difference between symptom presentation between men and women. And some of the people have found that as well, but our population is very unique, right? One of the biggest problems is we don't have enough women. So we have to really be careful about the end. So we try to collaborate with all kinds of different, communities and VA hospitals and also universities so that we can increase that number of people. So we can have equal number of men and women. So we can do not equal, but a larger number of men and of women. So we can do a better analysis. So, of course, I study the brain. So I wasn't just going to talk about symptom presentation and functional outcome. I have to talk about what their brain looks like. So we have this also under review in 2 other journals, different versions of this. And so women have thicker cortex before injury. Women, on average, now cortical thickness is a really interesting way of looking at the brain. That means about, I don't know how many centimeters it is, and it should be about maybe 2 to 3 centimeters deep. So if you took your brain, and this is going to sound a little bit B-grade horror movie, but if you took your brain and you kind of take a knife and cut like the centimeters, and then you flatten it out, you're looking at the cortical thickness. Now that cortical thickness actually changes from birth all the way to in your early 20s. It kind of goes up and down, and then it flattens out, and then it goes up and down. But it's something that is pretty much stable at and changes with age, but gets to be stable around in early 20s. Well, if you do look at the literature, women generally, or female brain generally has a thicker cortex than men. After injury, what happens is men tend to come down to their normal level where they were, but after injury, women still remain thinner. They don't go back to what they were. So there's a definite change in BHAP, like where they were before versus where they end up being after brain injury. So again, the difference is now translated after injury, and that should really help us understand how to take care of them, because this is not happening in the men, it's happening in the women. So this is like just like telling you different ways of analyzing data, different ways of looking at what is happening around you in between your parents, men and women, between you and your sister and you and your wife, and this is your way of understanding from behavior to the brain. Impact on everyday social interaction. Well, what does that mean? It means now we get to put everything together. We must think of brains as trees on a windy plain. The biology helps them grow upwards, but it is the wind that shapes them and makes them grow more branches on one side. So we all know we have asymmetry. When sometimes your left foot is bigger than your right, or your left hemisphere is bigger than your right. So you have asymmetry. You have also differences in the way your brain is connecting, in the way you're experiencing behavior and exhibiting behavior. But how does this impact our social interaction, and how should we address it? Should we think about it in a categorical way, or should we really think about it as a continuous way? Well, this is the age of artificial intelligence, it's the age of machine learning, and it's the age of understanding human factors. And the best way to understand human factors and combine artificial intelligence and machine learning is using the brain as a substrate. And that will translate into these differences exist and they must be studied. But the numbers must increase in research for us to know more. The power that's attributed to biology needs to be challenged. And the way it needs to be challenged is because we now have the tools that we can challenge it with. We don't just have to say, oh, I put an electrode here and look, it just went like, no, you have lots of prediction power, you have lots of data from before, and you have a lot of understanding of health disparities, healthcare disparities. You know that genes from having genetic predisposition to Alzheimer's is going to put you on a different cognitive track. You know that genetic predisposition to cardiovascular disease, cancer, or breast cancer, or Alzheimer's disease, or having the Alzheimer's gene is going to put you on a different track, not just with your heart, but also in your brain. So you know that you can integrate these things to understand how people will either have disease or how they will grow old or how they will perform in society. So there's a lot that needs to be challenged just in biology. And I'm saying that as a neuroscientist who grew up believing that biology is the best subjects that you can study. So there is no weaker or inferior brain. Obviously, we know that. We don't need to say it. We need to obviously pay attention to how our experiences drive our brain, obviously, in like a big, huge way. The father, this is a very good example, the father of a newborn baby who's the primary caregiver of the baby will have much lower testosterone level than a father who's not a primary caregiver. So I'm telling you this because this kind of research tells you about what we are in terms of a mosaic. Depends on our experience. So the mosaic model is something that integrates environment and biology. And that's why I've been talking about it in terms of this. So people who like these models and are going to be very interested in this, and you can read about this. Most human brains are composed of unique mosaic of features. And they conclude with this study that I'm talking about, it's from 2015, that human brains don't really belong to two distinct male or female, but that one sex category just provides very little information about the specific composition of one's unique brain mosaic. The study that I'm talking about is Joel et al. The present study, it supports these conclusions by saying that even when biological relevance is ignored, the structure of the human brain does not fit into two distinct type of brains, that one is typical of male and the other is typical of female. It's possible that although it is possible to use one's brain's architecture to predict whether this person is female or male with accuracy of 80%, one sex category provides very little information on the likelihood that one's brain architecture is similar to or different from someone else's brain architecture. So some brain types that are typical of females are also typical of males, and large sex differences are found only in the prevalence of very rare brain types. So whatever differences they are on the whole in the population, they're very minimal, and the big differences happen in the exceptions. So a rare type of brain can also be through machine learning in this model can also fall into the female category and also a male category. So I think that is a really important point because it provides you with something that I like to call the mosaic model. The brain types typical of females are also typical of males, and large sex differences are found only in the prevalence of some rare brain types. And this kind of brings us closer. It kind of brings us closer to the whole continuous form, and it brings us closer to, yes, we're seeing these differences, and yes, they're being they're affecting social interaction, they're affecting leadership, they're affecting health impacts, and we need to study them and make sure that the health precision models really do address these issues. But the fact that this kind of differences happen in the population, and the fact that the way that our atypical brain can also be atypical male, help us understand that these differences are not low or high or, better or worse. They actually help us strengthen our understanding of the human brain and help us understand in various ways on how we can help human beings. So if because of these deep intricacies that we have found, we can do incredible precision medicine. So this is a very mosaic model taken from the mosaic perspective into this. So you have, and this is very, very relevant now with the COVID-19 stuff that's coming out. So you have education and social factors. A lot of people call them SES, social economic factors. Then you have social economic status is what it's called. You have age and income. These are highly correlated, right? You also have race and ethnicity. So a lot of these also blow into the environmental, into genetic factors. What were you born with in terms of proteomics? What your disease factors are genetic? What kind of product are you going to create, which is proteomic products? And how that leads into overall function and overall behavioral outcome and what your brain health is. So all of these differences can interact together and create a continuous, and what we end up getting is a cross-sectional picture of what an individual looks like. And we need to make sure that we get all the factors in there and account for them and provide them the treatment or the rehab that they need. And so the mosaic model, I love this mosaic. It's there's no male or female brain. That's what I look at. The role of culture is not zero, and the role of biology is not zero. There are differences, yes, and we should address them. But at the same time, as we go along collecting the type of data, you notice, with the men and the women, and we target our medicine with knowing these differences, we will figure out that there's a lot that happens in the middle, and there's a lot that happens on the sides, and it just allows us to do better precision medicine. So, and it's continuously evolving because brains are plastic. So age will affect it, education will affect it, disease will affect it. And this is very important to understand because there's the diversity in neuroscience is needed to understand this. So as a scientist, I'm a basic neuroscientist, and I work with people from all different domains. Because in order to do this kind of work, you have to work with physicists, engineers, biochemists, biostatisticians. Now I'm working with infectious disease, virologists. Because of COVID-19, we work with a ton of people, epidemiologists, psychologists, computer scientists, in order to figure out, you work with your students, you work with psychiatrists, neurosurgeons, and you do this because you want to make bridges. Because you work in a mosaic, and that's what your brain is, and your environment is also a mosaic. So it is a universe within itself. And I think one of the best quotes that I've heard in my life is when I visited, I was a faculty diversity seminar at Stanford, and I heard a woman astronomer talk about the kind of research that she does from the Hubble telescope. And she talked about how one paper from their data has like 35 or 45 or God knows how many authors. And she said, we never have any first author, second author, or last author fights, because that is ridiculous. Because we work in understanding the universe, and you need a ton of people to understand the universe. And in my mind, if the closest universe to us is our own human brain, and it requires a ton of diversity in order to understand what's happening, and of course to provide tools for us to deal with the daunting obstacles that are those that are inside the heads of the people we talk to. And these are the take-home messages. Differences in the brain and behavior do exist. They impact health and social perception of both men and women. Despite previous sexism in science, advanced techniques reveal a brain that is neither male or female and shaped by the environment, both at the macro and micro level. And we have the mosaic model, and we need to harness this diversity for a successful career in parenting, relationships, and nation overall. And that's it. I hope I didn't go over time.
Thank you so much, Dr. Adamson. That was a fascinating presentation. Opened our minds like different aspects of neuroscience, the male and female brain. We do have a few questions in the chat as well. I can read them out if that might be easier for you.
Let's go ahead and share it because it's on the other screen.
Yeah. So the first question is from Miss Mahathi Rajkumar, and the question was, does the ratio of the weight of the brain to body weight matter?
So that's the body mass and the body mass index. So when they do look at the size of the skull and the brain inside, they do look at the body weight as well. More so, they also look at the amount of blood that's flowing. So the old way of saying it is that men have 1 liter more blood than women. So yes, all of those things do come into.
The next question is from Kushbu, and the question was, do you believe technological devices have an impact on these sex differences on mental health? And if so, how?
So I just submitted a paper that is accepted, and it's called Sex Differences in Treatment in Clinical Trials. So I can send it to you. So yes, this is a very important question, and nobody has written about this. I mean, this is like insane. Like, I cannot tell you the low-hanging fruit we have here. I mean, it's just insane that nobody's done this. So what happened about a year and a half ago, this is a very interesting story. I was at a conference, and the conference had a smaller women's gathering to increase diversity for women. I always go to those so I can just, stand there and laugh. And it's really interesting to me that, they were talking about how they can have all these women, but the actual conference was about devices. And most of the clinical trials that use devices, if you stimulate the brain, so a big part of my research that I do is neuromodulation. So I do repetitive transcranial magnetic stimulation. So I stimulate the brain. So I don't do deep brain stimulation. I do these huge magnets on your head, and I do clinical trials. A lot of the times that you do these treatments, you look at the clinical trials. In their papers, they report, number of females this, and that's it. They don't talk about how it affected them or anything like that. Well, there's a little bit of work in this that has been done. Few studies that show previously that depending on what time in your menstrual cycle you are in, the treatment effects are going to be different. So if you're in your luteal phase versus, depending on your menstrual cycle. Also, if you've had menopause. So the way that the magnetic stimulation is going to actually dissipate in your brain once it crosses the skull is going to be different in women, different times in their cycle versus men. And this is just 1 or 2 studies that came out. So we ended up writing a review paper on this, and we ended up looking at all these clinical studies, and we're like, they're not even talking about this. So we pointed it out, and that's a great question. So yeah, I hope that answered it, but I'm very excited about our paper that got accepted.
The third question is Ishav, and it was, in one of the slides, you mentioned that the white matter tract in the MRI scan shows the flow of water in the brain. How does it depict that?
So I actually did not, this is kudos to you guys. I mean, this is like Society for Neuroscience, right? So this is, and I noticed my background. So this is the virtual background from Society for Neuroscience from their 50th anniversary. And what it's showing is the rat hippocampus. So it's neuroimaging that actually is picking up the cells that were discovered by silver staining about 150 years ago by the father of neuroscience, who is Ramon Cajal. He's a Spaniard. And you can see from 150 years ago, when we used to use silver staining in like slices of brain, we've gone from there to neuroimaging, where you can actually put people in the scanner, make them listen to these really loud noises, and you end up getting what you get an MRI scan, right? You get your gray matter and your white matter. What you can do is changing the different frequencies at which you acquire the images, and there's a lot of physics that goes in this. When you acquire the images, you can actually change the angle at which you acquire them and the frequency. And when you acquire them, you can get different types of imaging. You can get bold imaging, which is on top of the gray and white matter. You can look at how blood is circulating based on the oxygen, bold blood oxygen level dependence. It's a ratio of oxygenated versus deoxygenated blood. And that would give you those nice red blotches, the different color blotches, right? But there's a lot of statistics that goes into that. So that's functional MRI. That tells you whether a part of your brain is functioning or not. The structural way of depicting white matter tracks. So the white matter tracks are basically railroads in your brain. They're like fat bunches of fibers, right? Fiber tracks that go from, I mean, some of them are massive. They go from back all the way to the front. Some of them are more intricate. They're bunches of your axons. Everyone knows what an axon is, right? You've got a cell body and you've got this axon that actually is the one that transmits your neuronal signal, right? Action potential and all that. So I think what is really important is that so this is an example. The reason I'm into diffusion tensor imaging is because I study brain injury. When your brain shakes and you actually are in a blast, or you hit yourself, or fall on the ground, or get hit, your brain, when it moves, it can actually cause a tear on the axon. So your axons can tear, they can shear. And when they shear, axons are made up of water. So the water diffusivity changes. So the flow of water changes. And with the different frequency changes in the MRI, we can actually measure the flow of the water. And if we see a break in it, we can actually say, aha, that's where the injury is. Now, it's not as detailed as I mean, it is very, very detailed, but it's not as simple as I'm making it sound. It's very complicated, but it's awesome. And it can do all kinds of, so the picture I showed is about 20 different types of tracks, 9 on one side, 9 on the other side, and then 2 crossing. And that's it. So that's how your diffusion tensor imaging is created. I hope I explained it.
Yeah, thank you. That was really helpful. But sorry, due to lack of time now, we will actually go on to our next speaker. There are a couple more questions on the chat. So Dr. Adamson, if possible, would you want to respond to them maybe on the chat? That would be helpful as well.
Sure.
Thank you so much. And so now they're on the chat.
Okay, I'll answer. Go ahead with your next talk. Thank you.
And so our next speaker is going to be Dr. Gandhi. Dr. Gandhi, are you here?
Hi, can you guys hear me?
Yeah, we can hear you perfectly well. Awesome. So
okay, that's good.
We'll have Neva introduce you. So.
All right, so now introducing Dr. Avni Gandhi. So she has earned her bachelor's degree in biochemistry with biotechnology from St. Xavier's College and her master's degree in behavioral neurobiology from the Tata Institute of Fundamental Research, and lastly her PhD in neuroscience, sleep and circadian rhythms and biology from Caltech. On top of that, from 2004 to 2005, she was in Xavier's Research Foundation Fellowship, and from 2005 to 2008, she was in the Junior Research Fellowship Program at Tata Institute of Fundamental Research, where she was one of 8 recipients selected nationally from over 2,000 applicants. From this program, she also received the Best Master's Thesis Distinction Award, which is so amazing. And after this experience, from 2008 to 2015, she joined a graduate fellowship and assistant program at Caltech, where she was one of 16 recipients chosen for the graduate fellowship in biology. Through these years of experience, she has designed and tested bioanalytical and some cellular assays for transfer to CROs. She has also conducted research on sleep and circadian rhythms with a neurobiology approach at Caltech. And currently she works as a preclinical scientist at an early stage drug discovery and development company. So yeah, now that you have some background information on some of the amazing things that Dr. Avni Gandhi has done, I'll hand over the mic to her.
Hi, thanks for that introduction, Neva. I guess I haven't updated my LinkedIn profile in a bit, but I'm now working as at AstraZeneca as a bioanalytical scientist. So yeah, it's the next stage. Okay, so this is kind of my first time with the screen share thing. So let me just share screen.
And if there's any questions, would you prefer we ask them at the end as well? We can keep them in the Zoom chat. And then again, at the end, we want to address them.
Yeah, I think that should be okay. Um, yeah, I think, well, I'm okay with people jumping in and asking as I'm going as well. So that's fine too. Okay, can you see my screen?
I don't think we're able to see it yet.
Okay, let's try again.
There we go. Yeah.
Yeah? Okay. Okay. Okay, so messing with brains is kind of what it's a little bit of a jump from what you guys were just listening to. I was more into behavioral neurogenetics than I think the other speakers on your sessions. So I will talk about that. So I would like to first of all thank the organizers for organizing this event. I'm really impressed at your passion and initiative in organizing this event. This world is changing and you guys are riding out to meet it head on, and kudos to all of you. So as I understand, most of you are high school students and are starting your journey into the sciences with exciting paths and directions to choose from. So I wanted to share with you more my journey today with the hope that it helps you in your journey. And when you come to a fork in the road, maybe you'll think about some of these things. So following the Japanese principle of ikigai, the first phase of my journey was about figuring out what I love to do. So as a freshman, I was inspired by the work of Eric Kandel, who had just won the Nobel Prize for his study of the molecular mechanisms of learning and memory. I was intrigued with the simplicity of the idea that the physical form of memory lies in the strength of the connections between the synapses of the circuits. So the model organism his lab worked on was the Aplysia sea snail, and he studied basic reflexive behavior in the sea snail, which involved a tactile stimulus to the siphon, resulting in a withdrawal of the gill. So something interesting about his studies was when the stimulus is repeated over time, the gill is no longer withdrawn. And this reduction in behavioral response to a stimulus after repeated exposure is called habituation. So why is such a behavior useful to animals? Well, by filtering out such a constant sensory input, habituation enhances an animal's ability to focus on what is novel, what is important about its environment. So habituation kind of serves as a basic building block for normal cognition. So the next questions were like, how does this come about? Where is the information stored? What are the circuits? So work from Kendall's lab suggested that the changes were in the strength of the synapses between the different neurons. And I was intrigued by this and I wanted to know more. So in order to answer some of these questions, I decided to work with another organism and a different circuit, um, basically olfactory habituation in fruit flies, or Drosophila melanogaster. So Drosophila provided some advantage over Aplysia in that it has a very powerful genetic toolkit. So it allows, it was so amazing. You can actually express particular genes in particular subsets of cells at particular times, and I found that absolutely amazing. So also they have a short lifespan, so you can actually do experiments as fast as you can think of them. So and they have a variety of complex behaviors that you can work with. So olfactory habituation is what I chose, but there's aggression, there's phototaxis, there's vision, it's amazing. So we basically, it is known how in the brain a fruit fly processes these smells, which allows us to run experiments. So we have olfactory sensory neurons which respond to odorants, and in a combinatorial fashion they're activated in response to particular odors. And this information then goes into the antennal lobe where discrete sets of glomeruli are activated in response to this. So you can see here, this is where the glomeruli are situated. And these inputs are then modulated by a set of neurons, some inhibitory, some excitatory interneurons. And then this processed information is then carried from the to to the higher centers of learning and memory through projection neurons. So but the question is, how do you establish an olfactory habituation paradigm for fruit flies? So first, you need a setup to test their normal behavior. So I use this apparatus called a Y maze. Fruit flies have a tendency to climb upwards, so they're introduced into the setup from the bottom here. When they come up to the center point here, they have a choice to make whether to go into the left arm or the right arm. In this case, the left arm has in this case, the left arm has a flow of air, which is the control, and the right arm has the flow of benzaldehyde, which is a compound present in almonds. If you, it's like the smell of bitter almonds. So fruit flies do not like the smell of bitter almonds. So you can see that they're avoiding this in this particular setup. But if you expose them for like 4 days to this this odorant, then you will see the response decrease. So you can see here in white, initially they have a strong avoidance response, but after keeping them exposed to benzaldehyde for a bit, they have a roughly 50-50% response to benzaldehyde. So this suggests that there is a habituation going on here. So, okay, sorry. Okay,
Dr.
so
Gandhi, we had a quick question about what is a Y maze in the chat. So if you could quickly please explain what that is.
Okay. So the Y maze is basically what's shown here. So, okay. So this is an actual Y maze setup with the fruit flies in it. So this is how small they are. So you can see individual fruit flies in here. And this is the control arm, and this is the benzaldehyde arm. So this apparatus is known as a Y maze. Does that explain, does that answer your question?
Yeah, good explanation.
Yeah? Okay, thanks. Okay, so okay, so what I'll show you in the next couple, well, in the next slide, basically, I don't think I have enough time to talk about all of it, but is so in response to this olfactory habituation, we also saw a concomitant decrease in the volume of the specific glomerulus. So we tried this experiment with benzaldehyde and carbon dioxide. So carbon dioxide is basically when an environment is too crowded, you'd see a lot of carbon dioxide. So fruit flies are hardwired to avoid carbon dioxide. So what was really neat that in response to habituation to carbon dioxide, we also saw a decrease in, an increase in the volume of the V glomerulus, which responds to this. So this was one of the first instances of a volume change in a brain area in response to in response to a learning paradigm, suggesting that there was a major synapse and remodeling going on with this simple behavioral setup. So overall, my work and later the work of my colleagues who carried on the project after I finished my master's showed that changes resulting in habituation were initiated in a subset of interneurons, which I showed earlier, that modulate the output of the glomeruli. So those interneurons were responsible for the habituation, so reduction in response from that particular glomerulus, as well as basically not allowing that response to go ahead, allowing the fruit fly to focus on other odorants. So anyway, so that's one part of it. I should have given a reference to the paper, but that's fine. So going back to the next part of my journey, I basically, so I finished my master's. I was at a decision point. I was sure that I loved research in general and behavioral research in the neurosciences in particular. So I decided to figure out if I was good at it and also become better at it if I could. So I liked working with fruit flies, but wanted to work with organisms that took me closer to understanding human beings while still having genetic tools to work with to be able to understand the actual circuits. So with that in mind, I joined PhD I joined Caltech as a PhD student in David Prober's lab. So I decided to work on circadian rhythms and sleep in zebrafish. So zebrafish are vertebrates, which helps a little bit, and they're particularly amenable to genetic modulation. So zebrafish lay eggs that are transparent, so you can inject the eggs during the one cell stage and make transgenic lines, such as, so you can basically express GFP in a particular subset of neurons of interest and other transgenes. So in this case, I've shown the locus coeruleus, expressing GFP, which is a wake active center, and we use the same promoter to express other things as well. So I also had an example of an ablation line which expresses an enzyme that kills the cells it's expressed in the presence of a toxic substrate. So in that case, I used it to ablate the pineal gland, which is responsible for sleep. Also, while I was doing my PhD, it became possible to specifically target genes in the genome for frameshift mutations. So I used many TALEN-based lines during the course of my research, but one of them was this ANA2 gene mutation line. So ANAT2 is the gene that is the rate-limiting enzyme for melatonin production. And so on knocking it out, you see that melatonin levels are usually high during the night and low during the day. And by knocking the gene out, we saw a complete reduction of melatonin during the night. And obviously not much is expressed during the day. So then I could actually study the null mutations in genes of interest for the rest of my graduate career. So finally, one of the things that really attracted me to working in this lab was the possibility of doing high throughput behavioral assays. So one of the things that's difficult about studying behaviors is that you need the numbers. So behaviors are controlled by a myriad of factors. So statistics becomes really important when you're making conclusions about what controls the behaviors. So zebrafish in their larval stages exhibit a full range of locomotory and circadian behavior, and their small size makes it amenable to do like high throughput assays in 96 well plates. So I use these behaviors to study the circadian regulation of sleep. So hopefully this video works, and you will see this is what it looks like with zebrafish in a 96 well plate. And the video tracker instrument uses infrared light, so you can't actually see the light change here, but that was the startled response of zebrafish in response to the lights going off. So yeah, that's it again. So, okay. So on to the next slide. So circadian rhythms are, so I wanted to study the circadian rhythms and sleep, basically. So circadian rhythms are physiological, physical, and behavioral changes that follow a daily cycle. They respond primarily to light and darkness in an organism's environment, and they're found in most living things, including animals, plants, and microbes. Circadian rhythms are driven by biological clocks, which are an organ's innate timing device. So molecular clocks are a family of proteins that regulate each other's levels through feedback loops that result in a rhythmic rise and fall of their levels. And these feedback loops are affected by external cues such as light. So I was specifically interested in how the circadian rhythm regulated the expression of sleep behavior. Are there, okay, yeah, okay. So it's an interesting side story. One of the first scientists to study the genes responsible for circadian rhythm was Seymour Benzer, who was a professor at Caltech before my time. So the story goes that he thought to study circadian rhythms because of an observation that he made about his graduate students. Some of them would come in very early in the morning versus some of them would not show up before 11, but then would work till late. And so Seymour Benzer's group actually made the connection between genes and behavior, and they were the first group to isolate mutants in fruit flies that actually had different circadian rhythms than normal, and those mutants were then used to then figure out the molecular pathway that that circadian rhythms use. So, actually the Nobel Prize a couple of years ago was actually awarded to scientists to figure out the molecular workings of the circadian clock. So now the question that I usually get asked when I talk about zebrafish sleeping is, do fish sleep? Because you really can't tell from their eyelids. So an awake sleep and an asleep fish, awake fish and a asleep fish pretty much looks the same. So in order to study sleep, first of all, you have to define sleep. So sleep behavior has been defined by the scientific community as behavior that meets 4 criteria. It's accompanied by increased arousal thresholds, which means that animals that are asleep require higher stimulus intensity to evoke a response than awake animals. That basically means, like, if your classmate is asleep next to you, if you poke them, they're probably not going to respond. But if you poke a classmate that's awake, they'll immediately wake up and yell at you or something. But that's basically what arousal threshold means. So it exhibits, so also, and then the last part of it is sleep exhibits hallmarks of homeostatic and circadian regulation. This means that there's a proportional rebound sleep behavior following sleep deprivation, and that sleep is expressed at particular times during the day-night cycle. So that's the part that I was really interested in. So the two-process model of sleep regulation, which is a popular model in the field, posits that the timing of sleep and wakefulness is regulated by 2 processes, process S and process C. So process S, shown in blue here, is, so process S, shown in blue here, is a homeostatic process that basically responds to internal sleep need. So that is when you've been awake for a certain amount of time and you're tired, then you feel sleepy, and that's the homeostatic process. Process C, shown here in green, responds to environmental cues that determine the timing of sleep and wakefulness during a 24-hour circadian period, and is regulated by the clock. The circadian clock. So adenosine has been shown to be play an important role in mediating process S. So adenosine is the breakdown product of ATP, which is the energy currency that I'm sure you guys must have studied about. However, while the circadian clock mechanism has been characterized in great detail, not much is known about what connects the circadian clock to the rhythmic expression of sleep. So what are some of the candidates? So we thought that perhaps melatonin could be a good candidate for this process C output molecule. So melatonin is a hormone produced in many animals, including humans, rodents, and zebrafish, in anticipation of darkness. So it's manufactured in the pineal gland, shown here for humans and humans here and zebrafish up here on the surface. So rodents also express melatonin, and weirdly enough, rodent melatonin corresponds to the night, which is when rodents are at their most active. So rodents don't really make a good model to understand the role of melatonin. So melatonin is also tightly regulated by the circadian clock. Its levels peak at night. So that's shown here. And if you give humans melatonin, like exogenous melatonin, then melatonin can reduce the latency to sleep onset. So humans respond to exogenous melatonin, and but the thing about melatonin versus other sleep aids that you might take is that melatonin won't force you to go to sleep, and it can be broken down by light. So if you take a melatonin pill and then you sit up and keep reading, then it's going to have no effect really. So you actually have to take the melatonin pill and then turn the lights off, and then it might help you sleep. But they never tell you to not operate heavy machinery when you're on melatonin for this reason, because it's easily broken down by the light. So Anyway, so getting back to what I did, um, melatonin. So zebrafish are diurnal like humans and express endogenous melatonin. So we decided to generate zebrafish that lack melatonin. So we generated a null mutation in the enzyme ANAT2, which is rate limiting for melatonin synthesis. And as I showed you earlier, the mutation of this enzyme causes a drastic reduction in the amount of melatonin animals produce during the light. So we used a melatonin ELISA assay in this case to quantitate the amount of melatonin, and wild type levels are shown in blue, and the amount of melatonin in null animals is shown in green. So we then use this mutant to examine interactions between the circadian clock, melatonin, and sleep. So it's well established in many animal models that melatonin, oh, sorry, where am I? Okay, sorry. So we use this video tracker setup, and we decided to ask, is endogenous melatonin required for sleep? And we assayed sleep in these 96 well plate high throughput assays that I talked about earlier. So in this case, we define sleep as 1 minute of inactivity, because that has been previously shown by my grad student advisor as being as being equivalent to an increased arousal threshold. So we use this metric to quantify the sleep-like state. So we raise larvae in a 14-hour light, 10-hour dark period, and then load them into these 96 well plates, and then basically track their locomotor behavior for 4 days. For 2 nights and 2 days. So usually experiments generate usually experiments generate traces that I will show you here. And we can also raise larvae in darkness and then put them in the light, or raise larvae in light dark conditions to start up their circadian rhythms, and then put them in the dark and see what their behavior looks like without the masking effects of light. So locomotor activity and sleep traces generated by this assay look something like this. The left panel shows the average locomotor activity for wild type larvae, and the light and dark bars on the bottom indicate the day and night periods. And if you remember, like when I showed you the video, the fish have a strong startle response when you first turn off the lights, so that is actually captured here. So so the left panel shows activity, and the right panel shows the amount of sleep. And since zebrafish are diurnal, you can see that the amount of sleep that they show is increased is higher during the night. So I'm also going to show you the control groups that we used, which are the wild type and heterozygous sibling animals, which are important to include. And in this case, you can see that wild type and heterozygous sibling animals have normal amounts of sleep, but the blue ANAT2 mutant animals have a decreased amount of sleep during the night and an increased amount of activity during the night. So we quantitated this, and we found that ANAT2 animals have a 40% reduction in the number of sleep that they exhibit during the night periods. So from this, we concluded that endogenous melatonin is actually important for sleep. So the next question we asked is melatonin required for the circadian regulation of sleep? So we raised larvae in light dark periods to start up their circadian clock, and as you can see, the wild type heterozygous and the heterozygous and wild type animals basically have a normal circadian expression of sleep behavior. However, when we tested the ANT2 mutant animals, there is no rhythmicity in their expression of sleep at all. So basically they sleep at the same level, whether it's the day or the night. So this was, it basically decouples the rhythmicity, it decouples the sleep-wake behavior from the clock in the absence of melatonin. So while coming back to the two-process model, adenosine is the process S output molecule, but from our studies, we know that melatonin is the process C output molecule. So now to wrap up, I have, I think, 2 more slides left. Okay, so this work was published as a good paper during, yeah, okay, so as a good paper, but after this, I came to another junction point. So I had now figured out what I loved and what I was good at. But at the end of my graduate career, I was worried that all of this research we were doing wasn't leading to anything useful for human beings, at least not in my lifetime. And that worried me. So for the next part, I joined Aerovasc as I worked at Aerovasc for 3 years as a preclinical scientist to understand what the world needed while honing what I'm good at. So it was a small company that gave me the freedom to understand what the world needed and how it could be useful to humanity. And I learned how drugs are approved, what is the procedure, what kind of studies are required to take drugs from the bench to the bedside. And I learned how to work with rodents, which was a skill set that I completely did not have so far. And I worked how to, I learned quantitative chemistry as well. So also to point out, like I would have loved to join join industry and do neuroscience, but that was about the time when the clinical trials in Alzheimer's disease failed. So there weren't many jobs available in neuroscience at the time. So anyway, so finally, I started at AstraZeneca in February as a bioanalytical scientist on the basis of my MALBio work at Caltech, which I didn't get a chance to touch upon, and my experience in drug development from Aerovasc. And I found that this job actually fills 3 of the 4 parts of this, this circle. And I guess and I'm hoping that like as I learn more, I will be able to also go back to neuroscience, which is what I love, and I'll get neuroscience projects as well. But I guess as you start your journey, there's a bit of advice I'd like to give you. So definitely explore what you love and what you're good at, but keep an eye out for what the world needs and where the world is going. And so keep picking up skill sets that are useful along the way, and keep that in mind. So I wish you all the best, and I'd be happy to take any questions that you might have.
Thank you so much, Dr. Gandhi. That was a great presentation. Also, a lot of advice for us to take away from this presentation. Also, love the way you showed from Ikigai, right? Is that how you pronounce it, Ikigai? The way you took everything to what you love and what can help the community. And so we have time, I guess, just for a couple questions, and I'll read it from the chat. Keshav asked, one thing that affects the circadian rhythm is the presence of light on the body clock. What other signals are there for deep sea creatures or others who tend to stay out of the light?
That is a good question. So they have actually found, so there are circadian rhythms in other animals too. So food is another big driver of the circadian clock. So the presence of food actually can entrain the circadian clock. It goes both ways because the circadian clock drives when you feel hungry. Like if you've ever traveled to an international flight, you don't get hungry at the right times anymore. But if you eat at the right times, you can re-entrain your circadian clock. So that's one of the big ones. So light and food are some of the big drivers of the circadian, of entraining the circadian clock.
Excellent. And another question, this one is from me actually. So you mentioned the AANAT gene, right? And so how that produces endogenous melatonin. And so would it be possible, I guess, to kind of synthesize like the protein it produces, the gene produces externally, and then you kind of give it to patients, inject to them so that they could kind of recover, especially patients who are suffering from like sleep disorders, who are not able to sleep as well and not have showing more action and activity. Could that potentially be a way to alleviate some symptoms?
So you mean by injecting, sorry, one sec. I am I want to see you guys. Let me just end this presentation so I can focus on, how do
I think you can just press stop screen share at the top.
use meeting controls? Sorry. Oh, okay. There we go. That's better. Okay, now I can see you. I'm sorry. I like talking to people face to face. Okay, so so your question was whether you can inject something that suppresses the ANAT2 gene in.
Yeah, is there a way to like synthetically, now that we know that which genes specifically like the proteins will produce, which kind of are involved in this pathway, is there a way to synthetically manufacture these proteins, or kind of like a drug that could be used for injections to kind of combat symptoms of people having these like sleep disorders?
Well, I guess if what I would do personally would be to first measure their endogenous levels of melatonin. So it could be that I didn't talk about it, but there's many other things that also control sleep. So I would first measure which part of the pathway is causing the sleep disorders, and then maybe go into this. Unfortunately for melatonin, it's like very easy to just get over-the-counter melatonin and take it. Right. And that's much easier than actually messing with the genes in the human body in this particular case. So yeah.
Yeah, thank you for that. And so actually due to time, we might not be able to ask too many more questions. There are a couple more in the Zoom chat if we're able to look at it. I think Mahati asked one, and then Vaikunt also asked one earlier, like 229 if you scroll up. So if you would be able to answer those in the Zoom chat really quickly while we transition to our next speaker.
That sounds great. I'd be happy to.
And thank you so much once again, Dr. Gandhi, for this amazing presentation.
Thank you, and good luck to all of you.
Thank you. And so we'd like to introduce our next and final speaker now, Mrs. Vada Party. Are you there, Ms. Vada Party? Are you able to hear me?
Hi.
Hi, awesome. Great, you're able to hear me, right?
Yep.
So we'll have Harini quickly introduce you, and then I guess we can get started with your presentation.
Okay, sounds great.
Great. Harini, do you want to come on?
Yes, hello. So Aparna Vada Party graduated with a BS in neuroscience and computer science at Johns Hopkins University. She's interested in computational cognitive neuroscience and aims to understand how humans do computations that enable them to understand the world around them, such as object recognition. In her talk, she'll discuss neuroscientific approaches to cognitive computations.
Thank you so much, Harini. And Dr. Vadaparty, Ms. Vadaparty, would you like to share your screen?
Yep. Sorry, 1 minute. All right. So I want to start. Sorry, 1 minute. Okay, so I'll start off by saying that I'm not a doctor of any sort. I graduated from Hopkins with a bachelor's in 2018. So yeah, just Annapurna is fine. I'm not yet Dr. Vadaparty, although I hope to be one day. So today I'm going to be talking about careers in neuroscience and also like some of the cognitive neuroscience that I'm interested in. Yeah. Just to check, can everyone see my screen?
Yep, we're able to see your screen.
Yeah, okay, great. So, sorry. Okay, so what I'm interested in studying is object recognition. So specifically how we do it, and I'll get into it a bit in a few minutes, but what I'm hoping to do is kind of build neural network architectures using AI and machine learning to see if we can find out if the algorithms they are using are similar to the algorithms that our brains use to do similar tasks. So first, I'll talk a little bit about how our brains do it, at least what we know about it so far. So broadly, there is a dorsal and a ventral visual stream. So what that means is, so there's basically there's two visual streams. What that visual areas in the bottom or kind of temporal area of your brain. I know that you guys have studied various parts of neuroscience, so you're probably familiar with the 5 lobes. One of them is a temporal lobe, one of them is the occipital lobe, and those are kind of the areas that these streams go. So the ventral visual stream, sometimes known as a temporal visual stream, processes these things. So color, texture, details, shapes, and sizes, and is mostly for object processing. So that's mostly what I'm going to be talking about today. There's also the dorsal, that's more for space and movement, but there is of course some overlap. So yeah, those are kind of how your brain processes visual information. So it starts with V1, which is at the back of your brain. So this right here, the occipital lobe, is where visual information goes first after your retina, your eyes. And so what happens there is it mostly processes really specific bars of light, essentially. So if you see here, there's bars that are oriented at different angles. And so you have different cells in V1 that respond most to different angles. So for example, this cell, you can see these are spikes. So these are action potentials, each of the like little spikes on this neural activity bar here. So you can see that this cell has the most spikes for like bars that are at this angle. So here, these different angled black bars represent like stimuli at different angles. So you can see that this neuron, so it's the same neuron that's being triggered in each of these different trials. So you can see that this neuron has the most spikes for a bar that's at this angle. And so this kind of shows us that, like, okay, different neurons have the most response to bars at different angles. So that was done by Hubel and Wiesel in 1968, and this is like one of the pretty like famous foundational visual neuroscience experiments where we learned that kind of like the basis, the alphabet of the visual system, if you will, are oriented bars. So everything that comes next, everything that I'm going to talk about, everything that you'll learn about in any neuroscience classes you take, in terms of visual neuroscience, everything starts out with these oriented bars, and then the rest is your brain piecing together information based on the rest. So next, yeah, so sorry, a bit more about this. The different cells for different orientations are lined up kind of in columns next to each other in V1. So yeah, there's a columnar architecture, and yeah, you'll probably learn way more about this if you pursue neuroscience of any sort in college, but basically you have columns of left and right eye input. So these things that are green labeled L, I know it's kind of hard to see, but basically the green ones are input from your left eye, and then the light blue or white ones are input from your right eye. And yeah, you have cells that respond most to bars at different orientations. So you can see like this one is like straight up and down, and this one's a little bit angled, and then so on and so on. And you get basically every single possible 360-degree orientation. There's a cell for that, or a group of cells for that. Okay, so next, after V1, so I've put this little brain image on all of the slides so that you can follow along where in the brain we're talking about. So V1 is in the back here, and that's what we just talked about. It responds most to oriented bars. V2, what it does is for things like optical illusions, if any of you have seen shapes like this before, so like you kind of see these triangles, even though there's not actually an outline of a triangle anywhere, you can kind of like, your brain will fill in that triangle for you. And so V2 is the area that does that kind of thing. It takes the edges that it makes from having oriented bars from V1, and it takes those and kind of makes shapes out of it the way that we do in optical illusions. So next is V3, so that's over here. You can see we're kind of traveling from the back of the brain to the front of the brain. So V3 is kind of next in line for that. And what happens there is motion. So I've put this picture here. I don't know if any of you have seen this optical illusion before, but basically like the pink dots kind of, it alternates which dot is missing, and it looks like there's a blank space running around and around the circle. And so V3 is for motion. And I was just putting this as an example because it does motion in the sense that like we think of it with just, you know, an object moving across the screen like my hand is doing right now, but it also does kind of these more optical illusion-y type motions. So it'll be the thing that interprets, like if you gave a computer just a picture of that blank space moving, it would say, oh, this isn't several images with a blank space moving, whereas your brain will say, oh, there's a like one thing that is moving in a circle, as opposed to the computer saying there are several images that have a different area of blank space. So I think that's an interesting example of how your brain kind of pieces together things holistically in a way that we haven't really done via artificial intelligence yet. So that's even at the pretty early visual stream regions of your brain, it's doing some pretty advanced stuff. So V4 is for color and shape. And so these are examples of some shapes that are preferentially recognized. So how I was talking about oriented bars in V1, V4, the kind of alphabet for V4 is this set of shapes, which they found kind of like by showing the oriented bars to in different positions and seeing which cells responded most to those. V4, they showed different shapes, and these were the shapes that different cells responded most to. And so it picks up on things like that and texture, as well as color. So next is V5, and so this is more about velocity, so specifically different directions. So whereas V3 was also for motion, V5 is more sensitive to objects going at different speeds. So when you're able to tell that one thing is going faster than the others, that's because you have different neurons that will have spikes at different, so different neurons will spike more at different speeds. And so that helps you determine which object of two is going faster, for instance. So those are all kind of the lower level. So I think specifically V1 and V2 are considered more low level visual processing. And then the last few I talked about were kind of mid-level. And then next we'll talk about more high level visual processing. So there are regions called the lateral occipital cortex and the fusiform face area. So LOC, FFA, don't need to worry about the names, but basically there are regions of your brain that specifically will light up for when you're seeing an object and when you see a face. And that's pretty cool because again, with the analog to computer programming, you would have to like explicitly tell the computer, here is how you recognize an object. And that is the kind of research I'm interested in doing. But it's kind of cool that like our brains are hardwired for objects, right? So you might think like, okay, that's not surprising, but it's actually pretty profound if you think about it. Like when you see a chair, how do you know a chair is a chair? Well, you could say, oh, it's something with 4 legs and this particular shape. But the thing is, you recognize it even if it's, you know, let's say behind a table or if it has 3 legs, right? You're still able to recognize those things as objects, even if it's not a specific object you've ever seen before. So it's pretty cool that your brain has regions that respond specifically to objects in general. And similarly, you have a region that does that for faces. So that's the fusiform face area. And I think some of the experiments for that are pretty cool. So I have a little bit more detail on that here. So for one of the famous experiments that was done is comparing faces versus houses, and how the fusiform face area responds more if it's looking at faces. So yeah, so okay, yeah, one of them was houses, another one was also just general objects. So here you can see they're comparing the activation level for faces and objects, and then intact faces versus scrambled faces. So a lot of times when you're designing an experiment, you have to be really clever in thinking of the control groups. So for instance, if I wanted to say, does this brain region respond more to faces than things that are not faces, you might have, like, the first thing you might think of is, okay, let's show someone faces and then objects and see if it responds more to faces. And it does, as we see in the first graph. So the bars labeled F are where the person was being showed faces, and then the bars labeled O is where they were being showed objects. And we can see that the fusiform face area, this little piece that is shown on the image, it has a higher percent signal for faces than for objects in all of the trials here. But that doesn't necessarily tell you that it's responding more to faces than objects, because it could, how do you define a face, right? Is a face just a set of eyes and nose and ears and mouth? In that case, we would expect that a scrambled face, so what you see here, it's kind of hard to see, it's not a super high res image, but here they have intact faces versus scrambled faces. So if all that your brain is picking up on is any pixels that represent eyes and nose and mouth and ears equals a face, then you would expect to see an equal amount of response for intact faces and scrambled faces. But actually that's not the case. You only see this kind of response. You can see I is for intact and S is for scrambled in this second graph. And so it has much higher response for intact faces. So what that tells us is, not only is it responding to faces over objects, but it has a very specific definition of faces. We don't know exactly what it is, but it's not just eyes, ears, nose, mouth. It is a configuration of those things. It won't recognize, like, this particular, the fusiform face area, not just me and Apurna, but like that specific part of my brain, it knows that there's something about a face that isn't just a set of facial features. And so that's really interesting. And the kinds of questions I want to ask are, how does it know that? What exactly is it picking up on? What is the algorithm it's doing? Because we've ruled out the algorithm of eyes plus ears plus nose plus mouth equals face. Clearly there's something more sophisticated going on. Is it maybe 2 eyes need to be next to each other and a nose needs to be underneath them? What about an upside down face? What about a face that is a color that humans don't come in, like blue? I think the answer would be yes, we recognize that as a face, because like when we see blue cartoon characters, we recognize those as faces, right? How would you do science about that, right? How would you design an experiment to see what exactly is the algorithm going on to recognize higher level things? So that's the kind of research I want to do. And if you notice here, a lot of the data that I've been showing you is graphs on an MRI or on a, like graphs based on MRI data or neural spikes. And that's a lot of what you learn about in a neuroscience class. But I personally, in college, when I was learning this, I was like, you know, this is interesting data, but that doesn't tell us what those spikes mean, right? It doesn't tell us what exactly those spikes are picking up on and what is being done with that information. And so as I studied more, I realized that the only way that you can really get answers to those questions is to build something that's predictive. So in science, we're always looking for theories that not only explain the data we have, but explain what you would expect from future experiments and we want to check that if your theory is right, it shouldn't just be able to explain past data, but it should be able to explain, oh, if you were to do this instead, what would happen, right? And so what I'm hoping to do is kind of build AI and ML models, so artificial intelligence, machine learning stuff with neural networks that some of you might have heard of. If not, no big deal. But basically building computer models that are able to have similar results to how humans behave. And so I'll say a little bit more about that. Yeah, so how do we get from low level to high level data? How do we figure out what our brains are doing to make that jump? Well, if you build a neural network that has activations similar to what we see, let's say on an MRI or on an EEG, and if it has activations similar to our brain's activations, and it makes mistakes similar to the types of mistakes people make in those tasks, then maybe the algorithm it's doing is similar to the algorithm that we're doing. And that's really helpful because we can pinpoint better an algorithm that a computer model is doing than an algorithm that humans are doing. Like, we can't just, there's a limited amount that we can see directly about algorithms that brains are doing, because brains are made of cells and we didn't program them, but we can program a computer and have a more direct understanding of those algorithms. So if we can make a mapping between what the computer is doing and what our brain is doing, then we can say, maybe the algorithm that the computer is doing is what we're doing too. And that's really cool, because now we have like kind of a set of rules or a set of patterns that we are using to do these tasks, and we can understand that through machine learning. So yeah, those are sources for the images. If you want, I can send them in the chat, let me know. So that's about it for what I had in terms of more technical stuff. Next, I'm going to talk a little bit about careers, and I'm really glad that the other speakers did too, because you guys are high schoolers, and I think it's really important for you to hear. I won't spend a whole lot of time, unless you guys are curious about me specifically talking about my career trajectory, but I wanted to say to you guys, you don't have to be a doctor. Most neuroscience majors, especially at Johns Hopkins where I went, were pre-med, and that can make you think that being a doctor is kind of the right thing to do, and it can be if you want it to be. It doesn't have to be. The Princeton Neuroscience Institute is one of many different sites where you can find a long list of careers in neuroscience that don't necessarily relate to medicine. So I'm going to spend a little bit of time talking about some of those. So if you are interested in the more technical and quantitative things, there are several options here. So it says investing in real estate, big data, AI, and brain-machine interfaces. So I told you a little bit about the kind of AI implications of what I'm interested in, like cognitive neuroscience questions, but there's also things like the brain-machine interface, which is really cool. And if any of you have heard of Neuralink, one of Elon Musk's companies, he's doing a lot of stuff with, or he and many other people are doing stuff with connecting computers to brains for things like prosthetics. So that's basically, there's a lot of like really engineering-y things you can do if you're into, math, physics, electrical engineering, those kinds of things, where parsing data from neurons, because what are neurons, right? They're sending electrical impulses. So that has really direct applications to electrical engineering. And so getting those signals, understanding what they mean. So feature extraction, what are the features being picked up on by these neurons, and pattern recognition. So what, again, kind of similar to what I was talking about, what are the algorithms, what are the patterns? And then using that to make a signal that does something with, let's say, a prosthetic hand, right? So these are really concrete things you can do with neuroscience and engineering. And of course, there's a million others. If you just Google neuroengineering, you'll find several. So also there's lots of implication for business and law. So in a cognitive neuroscience course that I took, actually, we had an entire kind of like seminar day where we debated different court cases that used neural data, and we said, was this a correct use of neural data or not? Like, did this prove what the legal entity, so like the judge and the lawyers, did this data back up the claims that the judge or lawyers claimed that it backed up? And so there's an increasing realization from the law field that, oh wow, we actually do need experts in order to be able to see if these things are saying, if this data is saying what we think it's saying. And you know, like, you can't really understand the data if you don't have this neuroscience background. So if you graduate with a degree in neuroscience, you know, definitely law is something where you'll be able to have an expertise that they're looking for. So if you love debate team, but you also love neuroscience, and you're like, debate team isn't going to help me be a doctor, you know, law might be something that you could look into if you love both of these things. And that's a very valid career as well. Yeah, so this is, you know, another example of that. It's being increasingly used. There's been lots of controversy over, you know, those lie detector machines, and I think most places don't allow them anymore. I think some places do, and it's bad. But so there are plenty of other neuroscientific tools that are used in court, and they're being used more and more. So people are definitely going to need you guys' expertise. Yeah, this is another example of that. There's an entire neuro law lab at Harvard. So, yep, friends in high places, I suppose. Then, yeah, so writing, there's lots of different ways that you can be involved if you're interested in writing. So scientific communication is an increasingly important field, especially with how interdisciplinary things are. Like, it's really hard sometimes to find someone who's well-versed in two different fields that are coming together. And I face this a lot in, you know, because I do things that are very overlapping between computer science and neuroscience. And a lot of times you'll actually hear, like, for instance, the more computer sciencey people saying, wait, but why do we care about this behavioral data? And then the neuroscience person will have to explain, oh, we care because it implies this constraints on the system we're building. Because, like, maybe the computer has some prior knowledge that, like, an animal living in the world or a human living in the world wouldn't have. So you can't assume that knowledge when you're writing your computer program. So things like scientific communication, super important. And then, of course, if you're creative, there are plenty of jobs out there too that are related to neuroscience. A huge one here is advertising. So it says, I don't see that specifically on this list, but I'll talk about it anyway. So I took a neuroeconomics class, and so there's a lot of different, like, kind of cognitive biases we have, and things like not wanting to take risky actions, so that's called risk aversion, that humans have. And there's just so many ways in which, like, art and advertising can leverage different aspects of, like, what is hardwired in our brain, things that you wouldn't expect a rational entity to do, because humans are not rational actors. Like, we are largely emotional creatures. We have a whole amygdala that makes us not rational actors. It's great. And so you can use that in advertising, and many advertisers do, and it can be very predatory and terrible, but it's also very creative and interesting. It's a terrible and wonderful field, but very interesting. So if you do like this kind of thing, it would be worth looking into. If you like thinking about how and why art makes the reactions in people that it does, neuroscience can be very helpful in explaining that. Great, so that's pretty much all I had. If there are any questions, I'd be happy to answer, and it can be about anything I talked about. So it can be about the technical stuff at the beginning, about the careers, about the college admissions process. I do also do that. I help kids with college advising as part of a mentorship and tutoring company. So if you have any questions, feel free to ask.
Thank you so much, Annapurna. That was really, really cool, especially from the technical aspect and talking about, like, all the various ways, I guess, like, neuroscience is involved. It doesn't have to be in medicine. I think that's, like, new for a lot of us as well. We've always thought of, like, neuroscience as, like, a strictly medical pursuit. So there's a couple of questions. One from Keisha first. This is about the technical part. So would damage to the LOC make it very hard for a patient to, say, identify a chair like if they, because I know you talked about the fusiform face area more in detail, but like let's say if there's damage to that region and they do see like a two-leg chair or something, how would that affect their behavior and their response to that new object?
Yeah, so the answer is yes, definitely. And it's good that you asked about like brain injury specifically, because in a lot of these experiments, the way that we figure out what a region does is by looking at patients who have injuries to those regions. Because I think the previous speaker mentioned she does TMS. And so that's like a thing where you can temporarily make a region not responsive, but it's for, and she mentioned it wasn't for like deep brain stuff. So there's a very limited amount of regions that we can do experimentally, like we can deactivate experimentally. So we do rely a lot for deeper brain regions on patients who have injuries there, because that's the only way that we can like have an example of what happens without that area. And so yes, patients with injuries in those areas would have a harder time with objects. And the answer to about the chair and like, is it harder to recognize objects you haven't seen before as much? It would depend on exactly where and how much the injury was. But yeah, you can see difficulties with different tasks based on which specific regions are deactivated. Yeah.
Yeah, definitely. That sounds like something that would have to happen, right, to understand how exactly the region functions, to look at like damages to the area. And another question I had, I guess, and this is not about the technical aspect, but since like all of us here are high schoolers, and we all seem to be interested in neuroscience, what are some things you'd advise for us to do during our high school years, so I get more interested in neuroscience, to learn more about the brain? Do you have any tips or advice?
Yeah, so I think that something really important you can do is indulge whatever you're interested in. So Google things, read things, even if you think it's above your skill level, just go into a Wikipedia rabbit hole, learn everything. Like, don't be afraid. Because I know in high school, I felt, even though it sounds really silly, I felt almost afraid to learn, because I was like, you guys might know that like this brain facts book, right? Like I thought like participating in the brain bee was the way to do neuroscience in high school. And I found myself like reading these things and being like okay, like some of it is interesting, but it really just feels like I'm memorizing a bunch of acronyms here. And like, it wasn't giving me a big picture, like understanding of what is actually going on in your brain. It was just telling me a lot of, brain facts, right? But that's not a holistic picture or a framework. So if you find yourself feeling that way, if you find yourself being like, I know I love neuroscience, why am I not loving what I'm reading? Google what you're actually interested in, right? It doesn't have to be in a specific like course or task. It can be just like, how do we recognize chairs, right? Or like, how does your brain know something is green, right? Or like, why does Alzheimer's make you have a harder time with this, right? Like, really just whatever you're interested in, Google it, read the big scary words, and then Google the big scary words. Like, I felt afraid to like even start with that process, because I was like, oh, like I won't be able to understand anything until I've taken XYZ courses. Like, neuro won't make sense until I finished AP Bio. And like, to some extent that's true, but to some extent there's a whole lot you can learn, and you can always like, you'll pick up those pieces of AP Bio along the way that you need in order to understand the neuroscience. And there are like everything I just told you, I explained to you without going into any detail about what an action potential is, right? And so like, there's plenty you can learn without having taken XYZ courses that you think you need before you can do whatever else. So basically, TLDR, don't be afraid to Google, and don't be afraid of your genuine interests, even if they don't align with what you're being told is the right way to do neuroscience. So even if it's not brain facts, even if it's not being a doctor, even if it's not whatever someone told you neuroscience means, that can be neuroscience too, and don't be afraid to indulge it.
Yeah, that's definitely helpful, because I know a lot of us are reading brain facts, preparing for the brain bee, and I guess that's the way we all prepare for neuroscience. So it's cool to like, I guess, follow your interest and your passion as well. Something that we all do. And then I guess the last question to wrap it up, this is from, or two last questions from Mahati. And so there's, the first one is, what good colleges are there for neuropsychology or neuroscience as a doctor? And what courses do you suggest to take if you want to be a neuropsychologist?
Neuropsychology. Okay. So I would not consider myself an expert in this. Once again, I would recommend heavy doses of Googling. But I would say neuropsychology is something that's like decently specific, so you probably won't find like a neuropsychology program per se, but you'll find schools with a strong neuro department and a strong psychology department, and then labs that are doing research in topics you're interested in. So I do happen to know that Hopkins has a very good neuro and psychology department, but it is also very pre-med, so depends what you're looking for. I also know that Washington University St. Louis has a program called Psychology, Neuroscience, and Philosophy, PNP, and that's a pretty cool program. I was looking at that when I was in high school as well. So that's, you can look into. But more generally, I think something really worthwhile doing, and this is something I did as a high schooler, is look at labs for every school that you're considering. Look at the labs there, see what research they're doing, see if it's research that you would be interested in. And if so, email the professors and ask them to have a chat. When I did this, there was not a pandemic, so I visited the professors and I had a chat with them, and it was awesome. Now it should be even easier for you guys, because all you have to do is arrange a Zoom call. Like, you don't have to fly across the country to do this. I kind of put that expectation on myself because I was like, I want to show them I'm serious. But like, now all you have to do to show someone that you're serious is have a Zoom call. So there's really no excuse not to do it. So if you find someone's research interesting, have a chat with them, like just email them. It's really scary. I can help with this again if any of you are interested, but it's just write up a few lines email. Do not copy paste the same thing to all of your professors. Copy paste most of it, but have a couple of lines in there that indicate what you're interested in specifically with that professor's research, and then email them and say, hey, I'd love to learn more about this, and then have a chat with them, see if you like it, see if you think it's a good fit. And then you can even write about that in your essay, in your admissions essay. You can say, I talked to Dr. So-and-so about this and I was excited about it. Like in your supplementary essays, when they say, why are you interested in this school? You can say, oh, I'm interested because I found so-and-so's research really interesting and they told me this when I talked to them. And that indicates that you're actually serious about the school because you went out of your way to talk to that professor. So that's something I think is valuable. But even if you don't bother with talking to them, even though I think that's a great idea, but even if you don't, just go and look at the research that different professors in that school are doing, see if you're interested in any of it.
Yeah, that's really helpful, especially for all of us now, or some of us starting the college admission process looking at that. That's great advice. Would it be maybe possible for you to type your email address into the Zoom chats, like if you have any questions or a way of contacting you, if that would be convenient?
Yeah, absolutely. I can do that. Let me just do that. It says to, okay, all right, to everyone. Great. So that'll be avtutoringoutreach@gmail.com. So yeah, you can feel free to email me there, and yeah, I'm happy to help. And we also have a team of Stanford and Hopkins and Princeton and Harvard college consultants. I think we might have hired more people, but basically I work with a team of very awesome other 20 somethings who are college students or graduate students, and we all do tutoring and college advising. So if you're interested, then we can set up a chat, and it's, yeah, I think we've been able to help a lot of people. So yeah, it'll, like, you can just chat with us directly, and then we'll have a chat that loops in your parents if you want it to become a long-term thing. But, yeah.
Sounds good. Thank you so much, Annapurna. And with that, that wraps up our presentations for today. Thank you again to everyone who attended for sticking with us through our technical troubles in the beginning. I hope you guys learned a lot from all these presentations, had many takeaways. And also hope you guys all signed in with the Google form that should be in the Zoom chat as well. But thank you once again to everyone who attended. Hope you guys had a great time. Hope you guys enjoyed their presentations, and we hope to see you in all our future events. So thank you, everyone, and thank you once again, Annapurna.
Yeah, thank you.
Yep, and that wraps up our event for today. Thank you, everyone.
Thank you so much, guys.